today, but yeah, thank you very much for joining us. I'm John Blair. I'm a postdoc in the Satija Lab, and I'm a member of this CICA consortium based here in New York City. So in this seminar series, we like to highlight new papers and methods from our center that we believe will be of interest to the broader community. So today we'll discover discuss two pieces of new work by Isabella Grabski and Tamara Prieto. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself to ask those questions, or you can also type them into the chat and I can read them aloud to the speaker. So our first guest today is Isabella Grabski. She's a postdoctoral researcher in the Satija Lab at the New York Genome Center. Isabella did her graduate work at Harvard University studying probabilistic methods for single-cell genomics. Her research now focuses on developing new computational tools for analyzing single-cell data, one of which she will tell you about today. So with that, Isabella, I will hand it over to you.
Okay, great. So yeah, thanks so much for the introduction. Today I'll be talking about a new computational method we've developed called RNA fingerprinting. So single-cell technologies are now exceptionally good at measuring how cells change across conditions. But we're still surprisingly limited in our ability to explain why cells are changing. So for example, what are the molecular drivers of those changes? We have tools like differential expression or gene set enrichment, but these are still fundamentally descriptive analyses. It's hard to look at a long list of genes and p-values or a set of gene ontology terms and actually understand what caused a cellular response. In our work, we instead identify a new opportunity for causal interpretation using a class of resources known as single-cell perturbation dictionaries. This refers to any data set that systematically profiles transcriptional responses in single cells under some array of perturbations. So for example, this includes data sets like genome-wide perturb-seq, which profiled thousands of genetic knockdowns at the genome scale, Skyplex, which characterized drug treatments across a range of dosages and cell lines, and of course, many other examples of perturbation screens under different biological conditions and contexts. Our goal is to use these perturbation dictionaries to enable causal interpretation of new data and experiments. So our key idea is that every perturbation can be thought of as leaving a fingerprint, that is, a gene expression signature on the targeted cell. If we can then match that fingerprint in a new context, we can infer what perturbation took place, giving us insight into what actually caused the cellular response. So here's a case study example of where this might be useful. Recent work has shown that many drugs, including those currently undergoing clinical trials, may not be targeting what we think they are in the cell. Our proposal is to match drug treatments to a dictionary of genetic perturbations as a way to infer their mechanism of action. So in this schematic example, for instance, you can see that the transcriptional signature of treatment with drug 1 looks like knocking down gene B. And analogously, treatment with drug 3 looks like knocking down gene A. So in both of these cases, we might infer these as the respective drug's targets. Beyond this example, there are many other biological applications, some of which I'll show today, where mapping data to perturbation dictionaries can give us causal insights that we otherwise couldn't have made. But first, in practice, this type of mapping problem is not as simple as it looks in this schematic. Real data is much noisier and high-dimensional. Here's an example of a real perturbation screen on the left, where I've grouped cells by these colors at the top according to the perturbation they received. Now let's say we have this group of query cells on the right that all got the same perturbation, and we want to map them to this reference to figure out which one it is. We have a lot more noise here than in our schematic from before, but in this case, at least by eye, we can identify that the cells received this perturbation. We can think of this signature, this pattern of gene expression that allowed us to make the match, as the RNA fingerprint for this perturbation. But now let's say we have this new set of query cells. This time, the signal is much less clear, and it really looks like they could have received any of these three perturbations. Closely related perturbations, such as members of the same complex, can have extremely similar RNA fingerprints. So an effective mapping method needs to not only account for noise and high dimensionality, but to also report uncertainty among closely related perturbations if we don't have enough confidence in any individual match. We address these challenges in our approach through a two-part statistical framework. First, we estimate these denoise perturbation effects, or RNA fingerprints, for every reference perturbation in the dictionary by using this multi-condition latent factor model. In particular, we model the expression of cells receiving each perturbation as arising from two main sources. First, latent factors representing baseline heterogeneity, like batch effects or cell cycle phase, that contributes to all cells in the data. And second, a rank 1 factor unique to each perturbation, which is what we call the RNA fingerprint. Each of these fingerprints can thus be thought of as a denoise vector describing how the perturbation affects the expression of each gene. By fitting this model, we're then disentangling these fingerprints from our unwanted sources of heterogeneity. Here on the right is a demonstration comparing perturbation effects between pairs of guides that targeted the same gene. You can see that conventional bulk summaries, like log fold changes, are correlated but noisy when compared across guides. By contrast, our RNA fingerprints are highly reproducible and show substantially less variability. Next, to map query cells to this dictionary, we model each query cell as a linear combination of these fingerprints. However, a naive regression approach would be unable to handle the potentially large numbers of highly correlated fingerprints. We address this by adapting techniques from statistical genetics. Specifically, in GWAS, it can be challenging to pinpoint the causal SNP for a trait because of correlation between nearby variants, which we can visualize using a Manhattan plot. When the causal SNP can't be identified due to this correlation, a so-called credible set of plausible SNPs, shown here in red, is reported instead. Analogously, in our approach, we can return a credible set of highly related perturbations if we don't have enough evidence in any individual match, and we can visualize this through our own version of a Manhattan plot. More generally, if we have multiple responses taking place in the same cell, we can return multiple credible sets, each corresponding to a distinct effect and describing its associated uncertainty. So, under this framework, we can summarize even complex patterns of uncertainty in an interpretable way. To now show RNA fingerprinting in action, we first illustrate our approach in settings with ground truth by using perturbation screens as query datasets. In particular, let's say we know what perturbation every cell received in one replicate, but we hide the perturbation labels in the other. Then the idea is we can treat our first replicate as our reference dictionary, and then map the cells from replicate 2 with the goal of predicting their true perturbation labels. We first tested this out with 2 replicates of a perturbation screen in hex cells, and then compared our results to the ground truth labels. Here in this heat map, every column corresponds to a credible set that we report for a different query cell grouped by their ground truth perturbation. For many perturbations here, we end up finding the exact right label with complete certainty. For example, all of these cells got an RPRD1B perturbation, and accordingly, we almost always report exactly RPRD1B. On the other hand, there are also perturbations, especially over here, where we instead tend to report credible sets containing multiple related members. For example, all of these cells got a CSTF1 perturbation, and we often find credible sets containing both CSTF1 and CSTF3. So when uncertainty exists, we can capture and correctly report it rather than confidently assigning the wrong label. As you can see, we also have a number of cells that we end up not assigning any label at all. And we find that this corresponds to heterogeneity and the underlying cellular phenotype. For instance, here, all of the cells in these first 3 columns received a CDC73 guide. The cells that we correctly labeled as CDC73 show a robust transcriptional phenotype, whereas the cells that we left unassigned in fact appear to have escaped the perturbation. In this way, our assignments can actually distinguish meaningful subpopulations. Similarly, we also find that different credible sets can also correspond to different transcriptional phenotypes. Here, all of these cells received either NUDI21 or a CPSF6 perturbation. For most cells, we are able to either assign exactly NUDI21 or exactly CPSF6, and those cells do show a NETI21 or CPSF6 specific module of expression. But there are also some cells over here where we instead report a credible set containing both of these labels, indicating uncertainty between the two. Those cells do show a more ambiguous transcriptional phenotype, and so highlight how our credible set framework can help summarize uncertainty. Finally, we also report a base factor for every cell that indicates our level of confidence in its assignment. As you can see, our correct assignments tend to have much higher base factors than our incorrect assignments, showing that under our approach, we can also successfully distinguish high and low quality assignments. We can also apply RNA fingerprinting across different biological contexts. Here, we try to map the exact same set of hex cells, but this time using a dictionary in K562 cells. These two cell types are quite different, and so for many perturbations like the one shown here, we have both common and cell type specific modules of expression, showing how challenging this problem can be. Nevertheless, considering our results across all perturbations, you can see we overall recover quite a bit of structure. We do find much more uncertainty in some cases, and there are some perturbations which are so different between these two cell types that we end up not finding any match at all. But when there is at least some common signal, we're able to drive a match even across these disparate biological contexts. So now what if we have multiple responses taking place in the same cell? Here, we applied RNA fingerprinting to a combinatorial perturbation screen, where we learned fingerprints for each of the single gene perturbations and then tried to map cells that received dual perturbations. Ideally, cells exhibiting two perturbation effects should map to two different credible sets, each corresponding to one of the true underlying perturbations. Here again, every column corresponds to a query cell receiving a dual perturbation grouped by their ground truth label. The different colors now represent distinct credible sets, and you can see we often recover the two credible set structure we hoped for, with each credible set in turn capturing its own uncertainty. Importantly, though, we still see quite a bit of heterogeneity, where in some cases we only find one of the expected effects rather than both. But we again find that this corresponds to heterogeneity in the transcriptional phenotype. Here I'm showing two examples of cells receiving dual perturbations split by their inferred assignments. For example, all of these cells received both an EP300 and an INS1 dual perturbation. You can see for the cells that were assigned as just EP300 or just INS1, we do see a phenotype consistent with that corresponding single gene perturbation. But for the cells that were assigned both labels, we instead see a much clearer combinatorial phenotype. So again, we can see how our fingerprinting assignments can actually help us distinguish substructure among the responses. So all of these dictionaries so far have involved anywhere from 20 to 40 perturbations. So now let's take things to the genome scale. This time, our reference perturbation dictionary is a set of over 4,000 perturbations from Genome-wide Perturb-seq, and our query data consists of over 1,500 perturbations from a separate experiment that only perturbed essential genes. Some perturbations here have very clear and distinct signals. Cat8 knockdown, for instance, has some shared signal, but also enough unique signal to give it a very distinct RNA fingerprint. As a result, for the Cat8 knockdown cells in the query, we end up assigning Cat8 alone with very high certainty out of over 4,000 possibilities. By contrast, there are other perturbations like MED30 that have nearly indistinguishable signals from many others. Unsurprisingly here, other members of the mediator complex. In this case, we find much more uncertainty among these different perturbations, and we end up reporting a relatively wider credible set containing MED30 alongside these other complex members. Here I'm showing more examples of assignments we make ordered by base factor, where you can again see a mix of both shorter and longer credible sets. You can also see that for our highest confidence assignments over here at the top, the right perturbation is almost always in the credible set, whereas for our lowest certainty assignments, we sometimes make related but incorrect calls. To summarize our results across the entire query data, here I'm showing the proportion of correct assignments, defined as having the right perturbation in the credible set as a function of the base factor. Overall, we make nearly 75% of these assignments correctly, and this only increases with our assignment certainty. So we're able to make accurate and meaningful mappings even in this highly challenging and massive setting. We can now return to our question from the start of drug target inference. Everything I've shown you so far has been between genetic perturbations only, but can we successfully map drug-treated cells to a dictionary of genetic perturbations and recover their true targets? To test this, we again used Genome-wide Perturb-seq as our reference dictionary, and our query data consisted of 5 positive control drugs from the Skyplex dataset. As you can see, for each of these drugs, we correctly recovered their true targets out of over 4,000 possibilities. Moreover, we even did so with complete specificity, where every credible set consisted of just the true target and nothing else. Under our approach, we can actually identify the genes that contributed the most to the base factors for these assignments. For instance, here, I'm showing the gene expression signature that allowed us to match one of the BCR-ABL inhibitors to BCR genetic knockdown. As a baseline, we also compared our results to using conventional approaches, like gene set enrichment based on differential expression, or cosine similarity of the transcriptional profiles. For this same example of BCR-ABL inhibitor, you can see that each of these approaches was able to nominate BCR as a potential candidate, but they also found anywhere from dozens to hundreds of other candidates, and BCR was never even the top hit. As a result, this ability to make a correct and specific match is a unique strength of RNA fingerprinting. We can also ask what our assignments might look like across different doses of the same drug. And in some cases, we do find a dose-dependent pattern of matches. Here, for example, you can see that this CDK9 inhibitor only matches to CDK9 at its 2 lowest doses, but not at its 2 highest doses. If we then look at the transcriptional phenotypes split by dose, we do see a very distinct response at these 2 highest doses. So our RNA fingerprinting assignments can also help us characterize a drug's dose-dependent selectivity profile. We next applied our approach to study 2 putative HDAC6 inhibitors. To give some background, HDAC inhibitors have long been studied as potential cancer therapeutics, but there's been growing interest in selectively targeting HDAC6 rather than broadly inhibiting the entire HDAC family. However, 2 such putative HDAC6 inhibitors, ACY738 and especially rigolenostat, have recently been suggested to not be as selective as originally thought. To test this, we treated K562 cells with these 2 HDAC6 inhibitors, as well as 2 positive control non-selective HDAC inhibitors across a range of doses. We then profiled these cells using FlexPlex, a recent technology from our lab that can simultaneously measure RNA and intracellular proteins in single cells. We then mapped these cells to a combined dictionary of genetic and chemical perturbations. The key idea is that if these drugs are truly selective for HDAC6, then they should map to HDAC6 genetic knockdown. Otherwise, if they act non-selectively across the entire HDAC family, we instead expect matches to other non-selective HDAC inhibitors. Here we can first consider our 2 positive control non-selective HDAC inhibitors. I'm summarizing the composition of the credible sets that we found at each dose, where you can see, as expected, that we either find no effect at low doses, or we find matches exclusively to other non-selective HDAC inhibitors. If we then consider our 2 putative HDAC6 inhibitors, we actually see the exact same pattern, where the only mapped credible sets are to non-selective HDAC inhibitors, never to HDAC6 genetic knockdown. This suggests that these 2 drugs are in fact not HDAC6 selective, and instead show a transcriptional response more consistent with broader HDAC inhibition. Because we also profiled intracellular proteins in these same cells, we had an opportunity here for orthogonal validation. In particular, we considered 2 canonical markers of HDAC inhibition, acetyl H3 and acetyl alpha tubulin. Under an HDAC6 selective inhibitor, we should only see an increase in acetyl alpha tubulin but not acetyl H3. By contrast, a non-selective HDAC inhibitor would be expected to increase both acetyl alpha tubulin and acetyl H3. If we first look at acetyl alpha tubulin under our two HDAC6 inhibitors, we see, as expected, an increase of this protein across doses. But we also see a very similar increase in acetyl H3 across doses of these drugs. This provides additional support beyond the transcriptional evidence that these two drugs act non-selectively across the HDAC family, highlighting the utility of RNA fingerprinting in identifying drug mechanism of action. Finally, we asked if we could apply RNA fingerprinting to study in vivo responses. To do this, we looked at the mouse immune dictionary, a data set in which nearly 90 different cytokines were administered into mice, followed by single-cell RNA sequencing of their lymph nodes. This generates a reference profile of how every immune cell type responds to every cytokine. By treating this data set as our reference dictionary, we can then map cells from any new experiment onto it in order to characterize what cytokines every cell is responding to. To illustrate this, we reanalyzed a recent study that looked at secondary immune responses in the mouse lung. The authors showed that after a first infection, populations of resident B cells form in the lung. Upon a second infection, these B cells then migrate to the sites of infection, resulting in this dramatic scattering of cells. What was interesting, however, is that in mice that had their alveolar macrophages depleted, we did not see this B cell migration. This suggests that these macrophages are somehow essential for this secondary immune response. However, the molecular mechanisms underlying all of this remain unclear. Because this study also generated single-cell RNA-seq data, we asked whether we could map this data to the mouse immune dictionary in order to identify what cytokines are driving this secondary immune response and how this might be altered in the absence of macrophages. Here I'm showing the results of mapping each B cell against this dictionary. And you can see strikingly that we saw three main categories of B cell responses. We saw B cells that were primarily responding to type 1 interferons, B cells that primarily responded to interferon gamma, and then B cells that exhibited both types of responses. This suggests that we actually have two axes of heterogeneity among these B cells. And indeed, looking at the transcriptional profiles, we see one trajectory of response towards the type 1 interferons and another trajectory of response towards interferon gamma. We then compared the proportions of B cells exhibiting each type of response across different mice. And what was interesting is the macrophage-depleted mice in blue showed lower B cells responding to interferon gamma, as well as to both types of responses. While of course the sample size here is too small to make any meaningful conclusions, what was interesting is the original study had actually independently and experimentally found a role for interferon gamma in this B cell migration, consistent with what we're seeing here. And so speculatively, we might hypothesize whether the macrophages are actually required for this interferon gamma response, and then subsequently the B cell migration. The last thing I'll highlight here is that under a standard unsupervised analysis of these B cells, we don't see any separation among these different response categories. And this is because each of these types of B cells is really differentiated by just changes in a small fraction of the transcriptome. This again highlights the utility of something like RNA fingerprinting, where we use supervised mapping to help us draw out these subtle but biologically meaningful effects. So lastly, I'm happy to share that both our preprint and our software are now available. With our software, we also include detailed vignettes, and we include pre-computed dictionaries for data sets like genome-wide perturb-seq in the mouse immune dictionary. And so I would definitely encourage anyone who's interested to please try it out. And with that, I'd like to thank my PIs, my collaborators, and my funding sources. And I'll also just especially highlight John, Alex, Carol, Jonsuk, and Isabella, who all carried out the experimental work that I talked about today. So thanks so much for your attention, and I'm happy to answer any questions from the audience.
All right, incredible work. Thank you so much, Isabella. So we'll move on to our Q&A. So if you'd like to ask a question, you can raise your hand or you can type your question into the chat box. I don't have a lot of experience with the hand raising, but I will do my best to find them. But in the meantime, I'll start with one. So Do you ever learn anything biologically interesting across different cellular contexts or something like that?
Yeah, that's a good question. So in general, we actually do find that if we map perturbations across distinct biological contexts, that gives us a way to actually find context-specific regulators. So I didn't talk about it today, but in the preprint, we actually show an example of mapping perturbations between two cell lines where one is p53 wild type and one doesn't have functional p53. And that actually helps us identify novel regulators of p53 under certain biological conditions. And so more generally, we can think of this as a way to kind of identify what perturbations are conserved between any two contexts and what might actually show some interesting biological differences.
Great. Okay, so I'll just give it a couple of 30 seconds or so. If anyone has a question, they can also feel free to unmute themselves and just ask it directly. That would be great. But another question I have, so a lot of this work is built on in genome-wide perturb-seq. There are other genome-wide perturb-seq data sets out there. Are you going to build dictionaries for them? Are they available in your software package for everyone? Or, yeah.
Yeah, a great question. So right now, if someone has any dictionary of interest, they can definitely learn the dictionary themselves with our software. But we're also starting to build this repository of pre-computed dictionaries. So I mentioned we have one for genome-wide perturb-seq and one for the mouse immune dictionary. And so as more resources like these continue to grow, we definitely plan to keep adding to that. And we're also interested in thinking about ways to make this more of a community effort too, so people can also contribute their own data and dictionaries that they.
Okay, great. Thank you so much. So if there's no more questions, then we'll move on to our next speaker. So thank you very much, Isabella. Okay, so our next speaker, sorry, I'm going to unmute her. Our next speaker, our second guest today is Tamara Prieto, a postdoctoral researcher in the Landau Lab at the New York Genome Center and Weill Cornell. Tamara did her graduate work in David Posada's lab in Spain, where she benchmarked different single-cell whole genome amplification methods and created tools for their quality control and reconstructed some of the first single-cell phylogenies of chronic lymphocytic leukemia patients. Her research now focuses on, her research, Her research now focuses on adapting phylogenetic methods to study somatic mosaicism in healthy tissues, which she will tell you about today. So with that, Tamara, I will hand it over to you.
Can you see the slides?
Yes, thank you.
Okay. So yeah, thank you, John, for the introduction. And hi, everyone. So first of all, I would also like to thank the Center for the opportunity to present our work. I'm very excited to explain the main results of our recent preprint, where we present the SMART-PTA, a method for joint single-cell whole genome and transcriptome profiling at scale that we use to reconstruct the evolutionary history of thousands of esophageal cells from healthy elderly donors. So its cell division since the fertilization till death of human individuals can imply one or more DNA mutations. And although we originally start our life with a single diploid genome, we end up with trillions of genomes in our bodies. So with time, we become genetic mosaics with tissues composed of many different clones. And along time, proportions of clones can change and new clones can arise in a continuous process that we know as somatic evolution. So phylogenetics can be used to understand any evolutionary scenario, not only species evolution, but also somatic evolution. As an example, the phylogeny of mammals reveals that dolphins are more closely related to cows than to seals. And the fusiform body shape of dolphins and seals are a phenotypic adaptation for aquatic life that is a convergent trait that evolved twice independently. So by calibrating this phylogeny, we can also transform branch lengths, originally an evolutionary rate, to represent time and reveal that their last common ancestor lived more than 60 million years ago in the Cretaceous. And the same principles can be applied if the terminal nodes are cell clones instead of species. And using phylogenetics, we can uncover how clones are related, understand and quantify their phenotype changes, or know when a clone originated, or even how fast certain clones might grow. So as this information can be of clinical relevance, how do we build the somatic phylogenies? We need to identify somatic mutations that arise naturally during cell divisions and determine which clone each mutation belongs to. Bulk sequencing can't always give us this information reliably because it infers clonal genotypes and abundances from variant allele frequencies. So an alternative is to isolate single cells and clone each cell in vitro to obtain enough DNA to build a sequencing library. And if cells cannot be cultured in vitro, we can still perform a whole genome amplification. However, historically, single-cell whole genome amplification methods introduce errors that make accurate phylogenetic reconstruction challenging. So tumor phylogenies have actually been built since 2010. However, healthy tissue phylogenies took a bit longer. And what you see on the slide is the first phylogeny ever built from hematopoietic stem cells and blood progenitor cells of a healthy individual. A landmark study from Peter Campbell's lab in 2018. And since then, more phylogenies have been built, some time-calibrated, but mainly from hematopoietic stem cells and hematopoietic progenitors expanded in vitro, as these cell types can be easily isolated by flow cytometry with very well-established markers, and protocols have been developed to grow them. But so far, we don't have any phylogenies of hematopoietic progenitors with higher resolution phenotypic information, only the cell type provided by flow cytometry prior to expansion, as the phenotype of the cells can also change during in vitro growth. And even more importantly, we don't have single-cell time-calibrated phylogenies of healthy solid tissues, or of tissues conform of mostly differentiated cell types, which cannot be grown in vitro. And for that reason, we aim to build a protocol that can be applied to virtually any human tissue, including, for instance, cryopreserved samples to reconstruct large-scale phylogenies with phenotypic annotations. And we chose to apply our new method to esophageal epithelium because this tissue represents a striking example of somatic mosaicism in healthy aging. It's similar to the findings in normal skin reported by Martin Corena in 2015 in Science, but this time in esophagus. And you can see that by the 7th decade of life, most esophageal epithelial cells carry pathogenic mutations in NOTCH1, NOTCH2, NOTCH3, TP53, and many other genes, but yet the tissue remains histologically normal and non-malignant. So a method that can be applied to any human tissue requires for direct single-cell sequencing at high depth, as we need to be able to genotype somatic mutations which arise in heterozygosity. But although single-cell whole genome amplifications based on exponential amplification introduce a lot of errors, more recently, a linear and quasi-linear amplification, such as primary template-directed amplification, or PTA, promise accurate genotypes, which can deliver more robust phylogenies. So for that reason, we decided to test PTA. And we built an in vitro evolutionary experiment by expanding DLD1 single cells during 4 months. And we PTA amplified and sequenced 16 cells, 2 from 8 different clones. And using the somatic mutations along the whole genome, we reconstructed a topology congruent with our design experiment. And importantly, all this note on the slide, which brand lengths correlated with culture time for each lineage. But the PTA protocol was really time consuming, so the team decided to automate it. And the major improvement was derived from the robotization of the protocol and the miniaturization of volumes in 384 well plates. And this way, labor was decreased to 2 weeks instead of the original 3 months for 1,000 cells. And the sequencing cost was dramatically decreased using ultimate genomic sequencing. And with the new modification of the PTA protocol from BioScribe, we can also obtain full-length transcriptions for the same cells. We then perform a quality control of 50 cells from the standard PTA plus RNA protocol versus 50 cells from our SMART-PTA protocol, and we show that our whole genome sequence at 15x provided coverage for around 90% of the genome with good uniformity, and our number of transcripts per cell was higher than 5,000 with comparable proportions of mitochondrial and ribosomal reads than a standard PTA plus RNA. So this way we obtain a cost-effective alternative. And now that we have a working methodology, we applied it to the esophagus. We first sampled a single esophageal region from one donor and did a standard PTA plus RNA. And then we applied a SMART-PTA workflow to biopsies from 4 different regions of the esophagus in the 3 elderly donors. And we flow sorted epithelial and immune cells, and we obtained more than 2,700 single-cell whole genomes with matched full-length transcriptomes. Using the RNA sequencing data, we detected multiple clusters corresponding to immune cells and also a big cluster of epithelial cells in different stages of a differentiation trajectory. And using expression of keratins and other markers, we classify epithelial cells into basal cells that divide into suprabasal cells, which transition to terminally differentiated cells before shedding into the esophageal lumen. We then quantify the number of somatic mutations per cell, and we observe that there is substantial variability across and within donors, all in their 70s. And the mutation burden was in general lower for the immune cells than for the epithelial cells within each donor, which agrees with the immune cells originated from hematopoietic stem cells that divide just once a year, while the epithelial cells derived from stem cells laying on the submucosa that divide each 11 days. However, T cells can expand sometimes within the tissue and divide significantly. What we think is happening in donor ESO3. So from these thousands of mutations per cell, how do we obtain a phylogeny with branches representing time? Building accurate phylogenies from thousands of terminal nodes is extremely challenging. For instance, in a study reconstructing the phylogeny of thousands of flowering plants, the authors use a divide and conquer strategy based on prior knowledge of taxonomic groups. But in our case, we don't have any prior knowledge per donor. So they also relied on a curated set of 353 nuclear genes known to be strong phylogenetic markers for flowering plants. But in contrast, we don't have in advance information of where the somatic mutations will occur. So they actually vary from person to person. So we need to amplify essentially the 6 billion bases of diploid single-cell genomes to capture enough phylogenetic signal. And using our matrix of somatic mutations, we build the phylogenies using a maximum likelihood method designed to deal with single-cell amplification errors, which in our hands work better than parsimony methods. And as you can see in this example, the immune cells are less dispersed as it is actually expected. And then we transform the branch lengths to time units using a relaxed clock instead of a strict clock to accommodate differences in mutation burden between immune and epithelial cells. And using a relaxed clock, we observe that the age of the most common recent ancestor of the germ layers, mesoderm and endoderm, agreed with the expected developmental timings occurring before birth. And this is the result for one of the donors. The phylogeny most likely traced back to the zygote or one of the first blastomeres. And we have sample cells whose most common recent ancestors lived at different time points of the donor's life. Then bootstrap support for the time estimate of each node is high, especially after embryonic development, with a global median of 90 out of 100 across donors. And as the internal nodes of the tree have been dated, we can group the mutations associated with those nodes in different time periods. So we decided to group nodes before 40 years old that we call early, after 40 to the last internal node or middle, and the most terminal nodes or late, corresponding to the cell sampling time when the donors were in their 70s. And after grouping them, we can plot their nucleotide profile. And as you can see, these two donors show different profiles, but also different time intervals show mutations likely due to different etiologies. And if we fit these profiles to cosmic signatures, we detect different clock-like signatures present across all donors and time periods, indicated in green and blue colors, but we also detect signatures SBS4 and SBS16 previously linked to tobacco smoking and alcohol drinking, only in the donors that had a history of doing so and during the periods that they self-reported it. For instance, in donor ISO1, the drinking and smoking signature is present in the middle, but absent before adulthood, and also absent from the latest period as the donor quit smoking 10 years before sampling. And after mapping the index ordering information, we noticed that immune and epithelial cells were not completely separated on the tree, although cell types still form distinct clusters, as shown by the significant positive autocorrelation and negative phylogenetic cross-correlation. Phylogenetic correlations are explained in greater detail in an earlier publication from our lab led by Josh Schiffman, but basically random distribution of phenotypes on a phylogeny, such our negative control, which is the percentage of a map reads per cell, results in a non-significant phylogenetic correlation, whereas a pattern structure will create a positive autocorrelation on the diagonal, and in case of two states, a negative cross-correlation on the antidiagonal. Immune and epithelial cells originate from the mesoderm and endoderm, respectively, but these embryonic germ layers are not monophyletic, meaning that they are not derived from single founding clones. So this explains why we don't see just two major clades, and instead we find early embryonic mutations shared between clones that give rise to both epithelial and immune descendants. And still, the presence of clustering is consistent with epithelial cells differentiated from lineage-restricted stem cell pools, leading to expansion of related epithelial or related immune lineages. And by sampling multiple regions along the esophagus, we can study migration of epithelial and immune cells. So high phylogenetic correlation means nearby cells are closely related, so little migration, while low correlation means more mixing. We found a strong spatial correlation for epithelial cells, but much lower for immune. And this matches their biology, as epithelial cells arise locally from tissue progenitors and are spatially constrained, whereas immune cells are motile and constantly replenished from the hematopoietic stem cells in the bone marrow. Taking a closer look just at the immune cells of this donor, you can see that, for instance, the clone highlighted in orange appears in 3 distinct regions separated by more than 10 centimeters. And the lymphocytes are resident, as indicated by the CD69 expression, and the clonal structure is supported by the shared alpha and beta T cell receptor chains in the transcriptomes of the cells within each clone. And then we decided to apply a phylodynamic analysis based on the accumulation of lineages along time. So smoothing the curves for immune and epithelial cells will reveal different dynamics. The more linear accumulation of the epithelial cells is likely due to continuous replenishment of the cells from the basal progenitors, while the hematopoietic stem cell population have been previously shown to grow exponentially before birth and stabilize throughout adulthood. And we only sample likely one descendant of each stem cell within the esophagus, except for the cases of T cell expansion. So this analysis can capture that biology. And then we focus on the dynamics of the epithelial cells. We selected the 2 largest epithelial clones, yellow and purple, with more than 50 cells each, and built again the cumulative number of lineages over time to reveal that clone A had a more steady linear accumulation of lineages, while clone B had a delayed but rapid expansion on the last decade. And after calculating the gamma statistic from Pybus and Harvey, we confirmed that clone B had an excess of recent branching, which could be related with acquisition of 4 somatic mutations in 3 relevant esophageal drivers that you can see on the screen. And as mutations in these genes seem to affect proliferation, we then look at all the mutations in genes previously interrogated in esophageal studies from Inigo Martin Corena or Seiji Ogawa. And we observed that frequent smokers and drinkers had a higher proportion of mutant cells than low-risk donors. And this was based on annotating clones with mutations in 28 genes previously identified as under positive selection. And we also found that TP53 and FAT1 are genes most frequently mutated across cells, the most frequently mutated in up to 9% and 4.5% of cells in some donors. However, NOTCH1 previously reported as the most highly mutated gene in esophagus was not well covered with PTA. These mutations in TP53 and FAT1 appear to affect cell phenotype. Mutant cells show an enrichment in more basal cells in comparison with wild-type cells that show more cells in suprabasal stages. And we then focused on all the TP53 mutant clones from one donor to assess the functional impact of this mutation. All non-synonymous exonic variants were predicted to be pathogenic with a CAD score higher than 20. And most clones carried the mutation in an heterozygous state, affecting only one copy of the small arm of chromosome 17, where TP53 is located. However, the clone carrying the most pathogenic mutation and about half of the cells from another clone showed loss of heterozygosity in chromosome 17p. This was clearly visible in the heat map as a loss of B allele frequencies from germline SNPs, indicating that the mutation in those cells had become homozygous. These mutations arose after adulthood, with most of them emerging within the last decade. So we then wonder if some cells express only aberrant TP53, the best known tumor suppressor, do these cells proliferate uncontrollably as tumor cells do? And when we calculated phylogenetic correlations of proliferation states based on the matched RNA profiles, only 2 of these TP53 clones with biallelic loss, the most pathogenic, which truncates the protein, seem to be positively correlated, indicating a growth advantage. But in contrast, the other clone in red seemed to even become senescent. So interestingly, too, a clone with a mutation in heterozygosity, the pink one, also showed a positive correlation with the proliferation states. And after doing a CMV analysis, we realized that only the 2 clones with the proliferative phenotypes showed copy number variants. The rest seemed to be euploid. And the presence of CNVs within some TP53 clones made us wonder if there were other aneuploidies in our cell population. And first thing we noticed was a loss of one of the chromosome X in the female donors, only in the immune cells, affecting from 20% up to 50% of the cells. And then focusing on the epithelial cells, we detected about 1.2% aneuploid cells based on read counts, which we validated observing allelic imbalance in germline SNPs. And this is likely an underestimate since we only counted CMVs present in 2 or more cells. And interestingly, these aberrant cells are still able to divide within phenotypically normal epithelium. And in one clone, we observe what appears to be an unbalanced non-disjunction event with multiple chromosomes amplified. Here, we can observe the same CNVs on the phylogeny, where we can obtain the age estimates of their more recent common ancestor. And it seems that all these aberrations have occurred in the 2 to 3 latest decades of the donor's life. And although most cells are diploid, we observed a much higher proportion of cells showing loss of heterozygosity in certain genomic regions in the first SMART-PTA donor that we analyzed. And this was evident from the B allele frequencies of phase germline heterozygous variants. Some of the allelic imbalances match copy number events we had already identified. However, other clones showed loss of one of the parental alleles without any detectable CMVs. For example, chromosome 17p shows LOH associated with TP53 mutations, as we discussed before. But we also observe LOH on 11q and on chromosome 22, the later carrying an EP300 mutation. And the most striking signal is on chromosome 9, where LOH is particularly widespread. And since we didn't detect CMVs in this region, we suspected that these are copy-neutral losses And importantly, they all include the NOTCH1 locus. So taking a deeper look at chromosome 9, after phasing the germline variants, we assign them just to one phase per cell to show that approximately 30% of cells in one of the donors show maternal and paternal allele losses. And as we suspected, the copy number state represented on the bottom row indicates that with the exception of a single clone, the remaining loss of heterozygosity corresponds to copy number state 2 in green, implying that these regions are copy-neutral loss of heterozygosity. And the fact that they affect different haplotypes and that have different breakpoints made us think that these events might have occurred multiple times. And we did an ancestral parsimony reconstruction, so we could confirm that the events occurred more than 30 times in this donor in these small biopsies. And so it is a trait that was acquired multiple times, similarly to how the fusiform shape of aquatic mammals I showed in the intro slides was acquired independently. So in this case, the CNLOH is all occurred after the donor was 20 years old. What made us also wonder if this was the case in the remaining donors. And after applying the same pipeline, we realized that CNLOH also occurred later during adulthood in the other donors, but the incidence of the events was proportional to the risk of the patients or the donors. With higher proportion of cells with CNLOH in high risk, then medium risk, and finally low risk donors. So this suggests that the CNLOH could be caused by mitotic recombination as a response to repair double-strand breaks, which are more common in donors than drink and smoke. But why the frequency of the chromosome 9q CNLOH is higher than the frequency of CNLOH in other chromosomes? We then took a look at the phenotype to try to answer this question, and similar to what we have seen for TP53 or FAT1, chromosome 9q CNLOHs might make cells remain in their most basal state and do not differentiate, and therefore increasing the frequency compared to wild-type cells that do differentiate and are shed to the esophageal lumen. And in fact, CNLOH cells seem to divide less frequent than wild-type cells, further showing the relevance of the cell bias mechanism. This is an explanation of the mechanism that could be causing the expansion of the mutant clones. And if this is the basal layer of the esophageal epithelium, some of these cells differentiate to maintain the outer layers of the epithelium in a process regulated by NOTCH1, but the signal pathway is not. And actually, the rate of differentiation can be interpreted as a death rate, as the cells that differentiate will eventually be shed into the lumen. A cell on the basal layer will then divide symmetrically to occupy the empty space. And during this division, a mitotic recombination might occur in a cell with an heterozygous mutation in NOTCH1. And this mutation will reduce the likelihood of the cell to differentiate. So a wild-type cell will differentiate to maintain the esophageal homeostasis, and then a basal cell will divide to maintain the tissue structure, and mutant cells differentiate at higher rates than wild-type cells. So after some time, the mutant clone will be selected. If we follow just some cycles, you can see how with differentiation, a bias in differentiation, in the end, we get like these clonal expansions that are so frequent in esophagus. And to summarize, we can now obtain thousands of single-cell whole genomes with matched transcriptomes for uncovering somatic mosaicism at high resolution in any healthy human tissue, even of those composed of mainly differentiated cells. And some of the new discoveries that I told you today include that calibrated phylogenies allow us to infer broad periods of tobacco and alcohol exposure. Essentially, a single sample today can tell us about past exposures, something that could be useful in forensics. And epithelial and immune cells show very different evolutionary dynamics. And while epithelial clones remain sedentary and are geographically constrained, resident T cells can migrate along anatomic distances. And more than 1% of epithelial cells in the esophagus are unemployed, but can still divide and propagate their CNVs to their progeny. And we also find frequent X chromosome loss in immune cells from aged females, similar to the Y loss found in elderly males. Some TP53 biallelic mutants can persist for decades without signs of cancer. showing that loss of both alleles doesn't always imply proliferation. And we also observe a strong convergent evolution of chromosome 9q involving NOTCH1, affecting different alleles and spanning variable proportions of the long arm. And these events arise through somatic recombination between homologous chromosomes, and their convergence strength increases with exposure to tobacco and alcohol. We think that mutations in TP53, FAT1, and NOTCH1 drive clonal expansion by biasing cells towards the stem cell compartment. And they don't make cell proliferase faster, but instead they block the differentiation. And in the context of epithelial architecture, that's enough to let clones expand. And crucially, we would not have discovered this mechanism without having matched genotypic and phenotypic information from the same cells. And finally, I would like to thank the whole team behind this work. First of all, Dennis, who is co-first author of the manuscript, for generating incredibly high quality single-cell whole genome data, and for all the great discussions about the CNLOH, TP53, the biology of the esophagus. Catherine for her writing, her creativity, the problems of solving skills, and the support, which helped us finish this project, I think, at least 1 year earlier. John for putting together a pipeline that we can easily run for each new donor and for parallelizing tree building on the cluster. Ivan for having the idea of the automation and for pushing us to measure and describe the trees properly. And then for the original idea of building large phylogenies, for making it possible and guiding us during the direction of the project. And I could go on forever, but about what each person contributed, but I'll stop here because of time limit. And thank you also to our collaborators, especially Julian Abrams and Akira Yokoyama. And thank you all for listening, and happy to take any questions. And I just wanted to leave you with this slide showing that as feeding on multiple plant hosts instead of having a single host during for the butterflies occurs multiple times independently during their evolution. And our data also reveals repeated independent acquisition of the same CNLOH event in different epithelial lineages along somatic evolution.
Okay, thank you so much, Tamara. Great talk. Thank you for sharing your work with us. We'll move on to the Q&A. So I wrote in the chat, if you'd like to ask a question, you can raise your hand or type your question in the chat box. We have a few questions already. So one is, did you look to see if mutations in non-coding regions had any effects on the gene expression?
No, we didn't focus on the non-coding mutations as much. Mainly because we didn't see like very huge differences in expression apart from the cell types differentiations, but it is definitely something that there is much more in this data that we should look into. It's very, very good question.
Great. And then just a clarification question. Is this based on cultured cells?
No, sorry. This is the SMART-PTA is just direct whole genome amplification of the sorted cells.
Okay, great. And then another question, could you give us a feeling for how many cells would have shown large scale, like arm or whole chromosome CNAs, if you had not required presence in at least 2 sister cells, and also considering the loss of heterozygosity events as well?
Yeah, that's a very good question. So before, previous studies have shown that around 3%, for instance, breast healthy tissue can have like CNVs. If you include, I think there are more mutations, no more CNVs present in our cells, but there is also, we just wanted to use the cells with the best quality for the CNVs, because I think the signal is clear when you have for the loss of heterozygosity, because you have multiple germline SNPs that you can use, but the signal from the read counts you have to correct well for the GC bias. And so including like I think probably this plot is the one that I had to go very fast in time. But mainly the plot with the heat map with the CNLOH, I would say that 40% of cells in some of the donors might have like might carry a CNV or a CNLOH. But in one of the donors that had like a high risk, in other donors we see much less. And that one, I think it would be, yeah, this slide would be like the most representative. And there are some biopsies that can get to more than 40% of CNLOH, but others with no tobacco and no alcohol risk, they actually have like only a couple of events in those small biopsies that we have.
Okay, great. Well, thank you so much again for the talks from both Isabella and Tamara. So this seminar has been recorded and it will be available soon on YouTube and also on our website, multimodalintegration.org. You can follow us on x.com at segs_ICA or check out our website, which will get back to updating for the latest announcements about upcoming talks and events. And so with that, I'd like to thank everyone for joining, and we hope to see you at our next seminar. Have a wonderful day.